So let's look at mitosis. Sorry, meiosis. Meiosis it is. So basically, meiosis is the division of sex cells and gametes, and basically this is your sperm and your egg in humans. Um, as with mitosis, the cells they are in interphase before division. Remember, that's sort of that optimal period of growth um, that's going on. DNA replication is going on. So it's getting ready for meiosis or mitosis to take place. So chromosomes will begin to replicate before meiosis happens. Basically, the easiest way, I guess, to look at meiosis is to do really a quick overview of it to start. So with meiosis, what happens, you have that DNA replication. And remember, we have 46 chromosomes. So if we have 46 chromosomes, here's a chromosome, here's another chromosome, here's a chromosome, and here's a chromosome. And you have those sister chromatids. And remember, they line up, or they sort of pair up with chromosomes that are very similar to themselves. So this is your homologous chromosomes. And just like with mitosis, it's the same phases. You have prophase, metaphase, anaphase, and telophase going on. So with meiosis, you have during prophase, the nuclear membrane starts to break down. It's after DNA replication, and they're sort of starting to pair up with their homologous chromosomes. In metaphase, so that's prophase. Um, in metaphase, actually, just let me go back here. Metaphase, what happens is they line up in the middle. The spindle fibers are attaching, but they're attaching to the homologous chromosomes. And then, so in anaphase, the ones that are being pulled apart are those homologous chromosomes. So it's not the sister chromatids. That's not until later. So what we have done, we have 46 chromosomes in here. So 1, 2, 3, 4. Let's just go with that for this example. Forget the humans. We have 44 chromosomes in the cell. And after this happens, if this were to divide into two, we have 2 chromosomes here and we have 2 chromosomes there. So we just went from a diploid number of 4 to a haploid number of 2. So we just cut those chromosomes in half, which is what the whole idea. So when we jump into the second part of meiosis, so sort of, cytokinesis is starting to happen there. So this is sort of, your telophase 1. And so you have your homologous chromosomes on this side, very similar ones on this side. The nuclear membrane kind of starts to reappear, but it doesn't fully, because what happens then when cytokinesis happens, this one is, going that way, that one's going that way. What's happening now, we have prophase happening again. Now you have to understand that DNA replication does not happen here. So this is not getting replicated, and instead, then what we're going to do, we're going to split these chromosomes in half. So you have your prophase, where they're kind of there, the spindle fibers are reforming. You have your metaphase, where they line up in the middle. Anaphase, those spindle fibers are attaching to the centromere. Remember, right here. Let me just make that a little bit different of a color so you can see it a little bit better. So, we have that centromere in the middle, and this is where these spindle fibers are now going to attach, and they're going to pull that sister chromatid apart right there in the center. So anaphase, they're getting pulled apart. And then telophase, you have the nuclear membrane reforming, the chromosomes are relaxing, they're not as condensed, and then you have cytokinesis, of course, happening, and that's happening with both this one and this one along those lines. So that's just the general overview. So what really you get is that double division happening. So you get the DNA, and that first separation, that first division of meiosis is separating those homologous pairs. First division of meiosis separates homologous pairs. That first division of meiosis separates homologous pairs. And then in the second division, this is when you have those sister chromatids being separated. So if we go through the steps, you have interphase, of course, happening. Then you have your prophase, metaphase, anaphase, telophase. And that first division of meiosis separates homologous pairs. So you're getting that diploid number being reduced to its haploid number, and that's why sometimes you will hear meiosis 1 being called the reduction division. And then once you go into meiosis 2, that's prophase, metaphase, anaphase, and telophase all over again. And then that's your second division, but remember it's separating just the sister chromatids. So it's just like in mitosis, you're not getting a reduction in chromosomes, because remember in mitosis, the whole point is just to replicate. It doesn't reduce any of the chromosomes at all. And that's what's happening in meiosis 2. So a little bit closer look, prophase, the chromosomes appear as single strands. You get those replicated pair of chromatids. So when you have that replicated pair, here's that, you have that chromatid here. Here's its replicated sister chromatid. And then it lines up with another very similar chromatid and its sister chromatid. So in there, basically what you have 4 sister chromatids lining up. And when this happens, this is now called that tetrad. And my pen doesn't want to work anymore. There we go, tetrad. So you have those 4 sister chromatids kind of there together. So let's just get rid of this. So then you have your metaphase happening. In metaphase, okay, those homologs, they are the ones that line up in the middle. So they're not actually separating the sister chromatids at the centromere. You're just pulling one homologous chromosome one way and its homologous chromosome in the other direction. So anaphase, then they're getting pulled apart, of course. And then telophase, basically your cytokinesis is starting. Oh, sorry. The cytokinesis is starting, and then you still have basically what you have now. Remember, you started with all of these together in one. So this is now, if we started off with 4, okay, so this was 4, we are now down to 2 and 2. Of course, if we're talking about humans, we would have 46, and then this would be 23 and 23. Meiosis 2, and here's the key part, there's no replication of chromosomes happening before meiosis 2. And that is key because now we're down to that haploid number, and all of the events are similar to meiosis 1, except now you're getting those chromatid pairs being pulled apart during the anaphase. So if you have this after meiosis 1, you get your 2 cells of homologous chromosomes, and there is no DNA replication. So now you get the homologous chromosomes are just, you have one set that's in this cell, one set that's in this cell. They're just lining up in prophase, lining up in the middle in metaphase. So in prophase, or the spindle fibers are starting to attach and they're starting to arrange themselves. Metaphase, they're lining up in the middle. anaphase, those sister chromatids are being pulled apart, and now you have a result of 4 cells. Remember that it originally started with 1, broke down to 2, and this, in humans, 46 chromosomes. If we just, use the numbers that we have here, this would have had 4 chromosomes. This one now has 2. This one now has 2. And those sister chromatids get pulled apart, and you now have your half sets of chromosomes. So we will continue to look at this in a little bit more detail, but I will leave that for another video.